So we have to talk about fish method. So yesterday we talked about microarrays, right? Before that we have been discussing about various electrophoresis methodologies. Of course, here are the methods which explore electrophoresis as one of its basic components in the entire process. But it is not limited to that. It is advancement of the technique for multiple applications. So here the first slide I'm sharing with you, which says ISH, FISH. Yes, these are the two short forms. ISH, as you understand, it's in situ hybridization. So it was very well early in the 19th century, 1969, by Purdue and Gall, as you are seeing in the slide. It was discovered, right? And of course, that time they had used it for study of 20s RNA that was hybridized to cytological preparation from Xenopus oocytes, right? So it was zoological study, but I mean the higher organism, not microbe, but it was to study the DNA or 20s rRNA that was hybridized to cytological preparation. So it is telling you it is about nucleic acids, right? Whether DNA or RNA. One more point, it is telling you about cytological preparation. That means directly cytology cells you are going to utilize in the process, right? So direct visualization of cell and direct visualization of DNA in the cell. So it is something different than what you just observe under the microscope, the cells, or you are talking about DNA, that is electrophoresis, right? So this technique is going to allow nucleic acid sequences to be examined inside the cell, inside the tissue, right? So integrity of the tissue or cell and its morphology, may it be entire cell or may it be its compartments like mitochondria or chloroplast, whatever may be, is going to be intact, integrated with this technology. You can visualize it, uh, the particular DNA sequence or particular RNA sequence expression also in a given tissue. So let's see what are the basic points in ISH, then we will go to FSH. It is a technique that allows precise localization of a specific segment of nucleic acid. Now, it is not about the entire DNA. We are talking about specific segment in the nucleic acid that can be visualized. So, and that you are visualizing in the intact cells. So, histological sections of any tissue you can check for. So, there is no need to extract the DNA from that tissue, but you are going to add your probed DNA to the histological sections and carry out the hybridization process, right? So here the ISH, when you talk about that of nucleic acids, is preserved adequately within a histological specimen. Histological specimen is tissue section, for example, and can be detected through application of a complementary strand of nucleic acid to which a reporter molecule is attached. That is what we call probe, right? So reporter is maybe a label, radio label, fluorescent label, etc. Now visualization, or maybe a chromophore, depends. So visualization of a reporter molecule will tell you about the mm, DNA or RNA sequence which is present in the entire cell population, and that probe will be finding the location of that particular gene sequence that you are concerned with. So in situ hybridization processes, when you talk about, the probes are of different types. Double stranded DNA can be used, single stranded DNA can be used. You can use RNA probe, and further you can use synthetic oligonucleotides, maybe short stretches or long stretches. And when we talk about labeling, here are the different labels, like for example, radioactive isotopes like 32P or 3H, etc. You can use other of alternative to this would be non-radioactive materials that can be used as probes, right? Now here the second component, ISH and FISH, that I had mentioned in the first slide. So ISH, you got to know that you are going to use some probe and you are going to detect a specific sequence in the histological section. That means direct at the cellular level you can release that probe, and that probe will go and bind to the nucleic acid which is inside a cell. It is not extracted out. Fine. Now when it comes to fish, what is the difference? Again, it is in situ different hybridization process. It is a cytogenetic technique that includes tissue cells as they are, but uh, third point is mapping of genetic material of the cell. Yes, same. So very much like ISH, only the difference is that of F, and F stands for fluorescence. So as we talked about ISH, similarly here, FISH is used to label the DNA by providing information on location, length, number of copies of specific gene or chromosomal portion. Right? So directly inside the cell, you can tap that. So this technique is based on specific interaction between the fluorescently labeled probe and a specific target sequence in the cellular DNA or RNA. So it is very much like ISH, but only the thing is the probe is labeled with a fluorescence molecule. So both FISH and ISH, the concept basically is same, that is in situ hybridization. Only the thing is the probe that are used in FISH, they are fluorescent probes, and that helps direct detection. So fluorescence allows visualization of probes in combination with the surrounding cells and tissues. So when you are looking for a particular DNA sequence in a given chromosome, then it is something like the example which I liked, and so I picked it as it is, that it says it is something like huge sequences are there when you talk about the entire genome in a cell, right? And you have to find out presence of a particular sequence which you are interested in, maybe a gene which is responsible for specific enzyme activity or protein production, or maybe a diseased condition, it is coming because of mutation in that gene. So from the entire huge content of some kilobase pairs of sequence, you have to find a few hundred base pair sequence. So it is something like finding a needle from the haystack, right? So that can be very well done by attaching markers to the oligonucleotide sequence, and that will be acting as a probe, and you can very well pick up the sequence of your interest. So detection is very direct and easy. So difference between ISH and FISH is only with reference to the fluorescence. Right. So as I said earlier, it is used to diagnose genetic diseases, gene mapping, identification of chromosomal abnormalities, and also be used to study comparison among chromosomal arrangement of genes of related species. So it has applications in systematics also. Right, identification and classification, positioning in the particular taxon or the taxonomical unit, OTU, that you can find out when you are going to take help of this technique as a molecular tool in study of classification. Now, whatever I am saying here, I will try to give some examples also at the end, like particular disease condition or gene mapping with particular references. So here it is just the summary of the technique which would help you to understand what it is about, right? So next comes the point, in situ hybridization. When I say I can use 16S rRNA also, right? Which is very much applicable in sequencing and identification and classification, taxonomy, etc. So, of course, as I said earlier, DNA, RNA, or PNA or LNA, oligonucleotide stages, they can be used. Further, you can use pathology when you talk about pathogen profiling, abnormal gene expression. Similarly, developmental biology, because certain genes get expressed at a particular stage of development of the tissue organ as per the age of the growing animal. So that also can be detected. So please mute your mics.
एवढं काय आता होतं ना त्या घरात घरात होतं म्हणलेलं ते
Please
आम्हाला
mute your
भेटतं
mics.
हे बघितलं आता कसं करतात आम्ही ते फ्रेम दिलं ना आता दोघी मिळून तर पैसे आता सांगते डायरेक्ट 10,000 द्या कसले पैसे
Rajnandini, please mute yourself. Okay, fine. Fine. So this is what we are talking about. So you can use it for 16S RNA as a target, and then it is for classification purpose maybe, or identification of a particular species. When it comes to pathology, applications are in pathological pathogen profiling or abnormal gene expression, It can be used in developmental biology, gene expression and profiling in the embryonic tissues or in the different developmental stages. Karyotyping and phylogenetic analysis. And the karyotyping is to nucleus, right? And phylogenetic again to classification. So this is what you can explore with the help of the techniques, both either ISH or FISH. Now here one formula I have written, but I'll not go into the details because it is technical about finding out the specificity. But yes, you can understand the concept, that is NMI, TNMI. These are the terminologies used. What does this stand for? NMI stands for the non-target microorganism that will not react with the probe. So, for example, you have a tissue sample infected with different types of bacteria, and you want to look for the particular Pseudomonas in that, which is first forming organism and the most what you say antibiotic resistant, that you want to find out in that tissue. So you have the probe which is compatible for that particular organism, and you have put into that in the process. Now some of the bacteria which are non Pseudomonas but also are present there, so this probe will be non-selectively not binding to those. It will selectively bind to only Pseudomonas DNA. So such case here Pseudomonas is target organism and others are non-target organisms. So NMI is non-target um, uh, microorganisms. So number of non-target microorganisms which are not going to react with the probe. And TNMI is the total examined non-target microorganisms. So when you are going to consider the sensitivity of the process, you have to take view of how accurately you are going to find it out. So what it is not going to react with, out of how many bacteria this particular target bacteria is identified, so on and so forth. So of course this is very technical, we are not going to go into the details here. I am trying to share with you the comparison of two techniques. So when I say fish technique, fluorescent labels, they're attached to probe and they're hybridized, right? So this is how you can see the fluorescence under the fluorescent microscope, right? But when it is about ISH, it is non-fluorescent technique. So you are going to use something like biotin. So biotin avidin or biotin streptavidin. This is what we had talked about, different compatibility molecules which are having specificity for each other, like in affinity chromatography. If you remember, we had enlisted a series of biological molecules which have biochemical compatibility for each other: antigen antibody, biotin streptavidin, so on and so forth, right? So non-fluorescent haptens like biotin or digoxenin or DNP, they can be added, or even a streptavidin or an antibody linked to an enzyme. So it is something like what you use in ELISA also. So it is not really new to you. So these can be the probe labels, right? So all these are non-fluorescent molecules, right? So can you observe the reaction happening? Can you observe that that probe DNA is going and binding to the tissue? Yes, as you can see here in the slides, it is observed under bright field microscope. Here you require, in the fish you require fluorescent microscope, but in ISH you require the bright field microscope. So this is overall introduction about fish. Now what it is used for, right? So here, as you must have seen, it is to localize a particular bacteria in a cytological section or histological section, right? Now here it is further going ahead, chromosome painting, that is another technique. Here you are seeing this segments of DNA and they are differently colored. Is it really possible? Is that DNA is painted like with different colors? Yes, this is possible because you are using different fluorescent probes and they are having specific fluorescence of a particular color. Yesterday we talked about green fluorescence and red fluorescence in microarrays and how you detect using that. So here also you are going to literally paint the entire genome by hybridizing the probes, and the probes are labeled with differently colored fluorescent, I should not say colored, but fluorescent labels, right? So multicolor fish is what will give you such kind of a picture, that is different probes will be binding at different locations, and each probe is having a particular fluorescent label. So colors of that fluorescence will be seen and the genome appears to be painted. So the technique is referred to as chromosome painting. Similarly, another application when you talk about that of fish is gene mapping on chromosome. Yes, as I said earlier, specific probe will go and bind at a particular locus of the genome, and you can map the particular gene on the chromosome. Also, it is applicable in analyzing the cells not currently undergoing mitosis. So mitosis is a particular physiological state of the cell. So metaphase, anaphase, telophase, all these steps, how they progress during mitosis. So this technique can analyze only metaphase chromosome, but fish can analyze both interphase and metaphase chromosome. That means the cells don't need to be cultured far in advance before chromosomal analysis. Of course, when you are talking about exposure of the chromosome, then and then only it can be tagged. So this is very very much important in I mean higher organisms, right? So because the DNA or the chromosomes, they are inside the nucleus. Nucleus has its membrane and it is a compactly arranged structure. But when it starts undergoing mitosis, then at a particular stage of mitosis, like metaphase, for example, the chromosomes will be properly in their expanded and relaxed form, and then it will be easy to tab them, right? So is it okay at metaphase or is it okay at interface? That is the concern. And if it is so, then you have to have the tissues in the particular growth phase, then and then only fish assay can be done. Fine. So when I talk about this, here the fish technology can be applied to both interphase and metaphase chromosomes. That means cells will be tapped. So whether it is a solid tumor or any infected tissue, that also can be tapped, whether the tissue is undergoing mitosis or not. So this is another example or of application that can be. Uh, I mean the data can be extracted related to uh, particular tissue or its physiological conditions, its infection condition, its disease condition, whatever it may be. So fish is applicable for this. Now I go to next slide. Here last application, just to summarize, karyotyping, that is what we talked, right? So nucleus ka staining ko bolte karyo staining, and analyzing it is karyotyping. So particular sequences within the nucleus, so it is referred to as karyotyping. So fish can detect deletions, translocations and duplications of genes. So that karyotyping will tell you about any mutations that have occurred, and you can correlate with the particular outcome of it, maybe a disease condition or maybe higher expression of a particular gene, etc., etc. So this is will help us to know about the chromosomal integrity. You can localize and measure the DNA or RNA content within the tissue. So gene expression again, so that will be tapped in terms of mRNA. So you can talk about the content of particular RNA in the given tissue, right? So you can get to know about the progression of the disease. You can identify the disease in its early stage. You can find out how it is progressing, and of course it can be utilized to check the interaction of drug molecules with the targeted sequences to inhibit them, to kill them, whatever kind of variations are happening as a result of treatment, right? So that also may be tapped with presence or absence of particular sequence that you are looking for. So this is overview of applications of fish. Now this is how you can see. This is the tissue section, and these small dotted lines, they are nothing but, right, you will see some arrows here, red color arrows, right? They are showing position of particular bacteria in that infected tissue. So here are the examples of the tissue which is infected with different bacteria and how that bacteria can be identified. For example, here it is proteobacter. In this case, here it is showing the presence of lactobacillus. Of course, not necessarily infection is caused by presence of bacteria. Certain useful bacteria are there in your entire system, so they also can be localized. Here it is ruminococcus that is identified. So as you can see, yes, they are seen as beautiful fluorescent structures, and you can identify the presence of that particular bacteria in the entire tissue. So your probe DNA will not bind to the DNA of the host, but it will selectively bind to the DNA of the uh, bacterial cell, and then it can be identified, it will get fluorosed. So here it is Clostridium species, so on and so forth. So here is your probe DNA. To the probe you have added the fluorescent signals, these yellow bright color ones, right? And then you are going to add it to the sample, and then fish probe will be getting interacting with the selective sequence in the DNA, right? And then it will bind with it, and it will give you the fluorescence. If it is not binding, there won't be any fluorescence. That is how it goes, right? Now the concern is how this DNA is getting entry into these tissues, right? So whenever any histological technique is performed, fixation of the tissue is one of the important steps. So now here we will be talking about these steps, but let's understand about the probe a little bit, right? So what is a probe? As we know, it is a complementary sequence, and this complementarity is towards what? To the sequence of interest, right? So the probe is fluorescently labeled by adding or conjugating the nucleotides with fluorescent marker. So yesterday we have seen some examples like CYN5 or CN3, etc. So the chromosomes are going to be mm, binding with this probe, right? So when I say how it is going to happen, so chromosomes are denatured. So denaturation of DNA, it can be done by changing the pH, temperature, or by adding the chemicals like urea. So maybe you are giving heat, or maybe you are adding chemicals, and the hydrogen bonds will break, and your DNA or the chromosome is denatured. So double helix structure is going to get unwound, right? And when you remove those factors like heat is removed or the chemical is washed away, then again DNA will resume its helical nature, we know that. So the probe is added to the denatured DNA. As you are seeing here, this particular zone is in denatured condition. The concentration of the denaturing agent, the buffer conditions, etc., will decide about the degree of denaturation, we know that. So it can be done under controlled conditions. So the particular DNA region is allowed to what we say a denature, and then the probe is added to the specimen for hybridization, and the specific binding to the complementary sequence will occur, and that is how you will see the fluorescence. Of course, excess of probe is to be removed, so washing is another step. Right. So once that probe is added, it will be interacting with the targeted chromosomal region, particular sequence. Excess of probe is washed off, and then that slide is observed under fluorescence microscope. Right. That is how the steps are. Now, to little more details. Now, probe design is a very challenging thing because you need to know the particular sequence which is going to be uh, functioning like the primer, right? Primer in extension of oligonucleotides. So specificity is very important. Similarly, here the probe should be very specific and truly complementary to the sequence which you are interested in. So the design of the primer is a crucial thing and needs lot of understanding about the gene sequence that you are playing with, right? Another thing is fluorescent labeling. The probes are labeled with fluorescent dyes, which allows the visualization under fluorescent microscope. So selection of these fluorescent dyes also is going to be important, specifically when you are going to go for technique like chromosome painting. Multiple fluorescent dyes you are going to use. Then in situ hybridization, this is what we talked about. I'm going to go to the next slide. I'm going to skip this. It is just blah, blah, blah. This is how it is going to work. So what you want to have the probe, you want to have the target, and the probe has to go and bind to the target DNA. But where it is not extracted pure DNA, it is within the cell, cytological technique it is. So you have to handle the cells in a manner that selectively the probe will go and bind to the DNA. Now here, the very first step, of course, is going to be fixation and permeabilization of the sample, because penetration of that DNA probe is to be there, and the tissue must be fixed on the particular slide. Otherwise, it will be very difficult to handle it. So fixation, if you remember, in the zoology lab you might have seen the animals are kept in the jars and they are in the fixed state. They are not going to change anymore. So that is nothing but you are using formaldehyde for fixation there. In in microbiological staining techniques you use certain fixatives also. So similarly here you are going to use fixative which is going to allow the processing of the cells, so they are going to be available for direct penetration of the probe. So cells are to be treated with the fixatives, and also permeability of the probe is to be allowed. So permeabilization, so you remember this term you had used in transformation experiment, for example artificial transformation. So what you do, you treat it with calcium chloride solution and you allow the cell membrane to open up in terms of the pores are enlarged and then the DNA enters, right? So permeability is to that. So the DNA, which is your probe, will enter only if the cell allows it to enter, and that can be chemically controlled by permeabilization by treatment with chemical reagents, which are going to allow the pores on the cell membrane to expand and allow the DNA to enter. This first step. Second is hybridization. So preheated hybridization buffer is required for this and added to the sample containing fluorescently labeled probes, which is complementary to target DNA. Of course, it has to be. So denaturation is one step, so which is carried out by adding the preheated buffer. So that will denature the DNA. Along with that, your fluorescently labeled probe also is going in that buffer, and then you are going to pull the system so that annealing of the probe to the target sequence will occur. It is very much like PCR or any other technique that involves denaturation and renaturation of DNA, right? Annealing of the DNA. So again, coming down to 37 to 50 degrees Celsius under dark conditions will allow um hybridization to happen, because this probe DNA will be binding to the unwoven or denatured DNA sequences. It has a possibility to find its complementary region and it will go and bind. So this is what is going to happen with the help of properly designed buffer system that is required. Then of course washing is required to remove unbound sequences, unbound probes, and then you are going to take it to microscopy. Or another instrument that can be used is flow cytometry, right? So this is how is in simple the steps of the technique. Now here it is very sensitive and specific method, that is what we expect, right? Instead of hybridization we are talking about labeling a particular DNA fragment, so it has to be very specific and sensitive method. Now what are the applications? One by one we are going to go into the details. So very first, as we said earlier and have seen the slide, it is for detection and localization of specific pathogen DNA in a sample using fluorescence or confocal microscope. See, by routine identification of pathogen in a patient, it takes 2 to 3 days, right? And it is very long period when the infections are uh, in a patient who are very prone to succumb the disease and are in a critical condition. So immediate high throughput method used to be there, which will give the identification of the pathogen, right? Of course it is going to be based on molecular tools, and PCR is one of such tools. Here is the another tool that can be used for localizing pathogen DNA in a sample. So the sample could be a tissue sample could be any of the what we say materials which are like say food sample, it is also going to have the cells, right? So food as in the cells of plant or animals, and presence of a pathogen in that, which is fluid pathogen, also may be detected using this technique. So here identifying the pathogen DNA in a given sample can be very well done, and so comparatively it is a fast and rapid technique. So is it limited to bacterial identification? No, as the last point covers, it is developed for detection of and identification of various viruses. So may it be viral hemorrhagic septicemia condition or viral nervous necrosis, whatever may be the tissue conditions which are caused due to presence of a particular virus. So this is what you can do with help of this particular technique. So detection of a particular DNA, that is what is the basis, right? So whether it is viral DNA or whether it is bacterial DNA, it is not going to be a concern. You have to have a right probe designed for it. So this is what we have already talked. Now I want to go to different examples. All right, this is how you can summarize the technique. That is the flow chart which shows fluorescence in situ hybridization. So you are going to prepare the hybridization targets first. All right, so pre-treatment and denaturation of the hybridization target. This is the tissue, for example. Then you have to do the fixation of it, right? Then denaturation with the help of treatment with buffer which is heated. Then to that you are going to add the the uh, the probe, and then denatured DNA, and the probe will bind, and you are going to get the hybridized molecule. Post hybridization wash is given, and this slide is then taken to flow cytometry. Fluorescence microscope. A sample in suspension can be taken to flow cytometry. So this is how you can schematically represent the fish technique, right? So you can use the locus specific probes, right? And what kind of signals you are seeing. Now in this particular case, what you are seeing is this particular tissue, right? It is chromosomal probe that is added, and what you are detecting here in this particular experiment, they have detected trisomy, that is presence of triplicate. Our chromosomes have are diploid, right? So two sets are there of alleles, but here trisomy is there, which is a rare and non-normal or abnormal condition. So this abnormality can be detected by fish, and that is how it is shown. So locus specific probe they are designed, and here it is whole chromosome probe that is designed. So whole chromosome design, whole chromosomal probe, and here it is locus specific probe. So that is how they are going to bind. So entire chromosome which might show the trisomy can be identified, and here it is particular gene within that chromosome which is having any abnormality or any variation can be tapped. So you will have different types of probes depending upon your target application. So locus specific or complete chromosome specific. So it tells you about the scale of work, right? It is not only a small piece of sequence that is only you can identify, but yes, you can scan a particular chromosome in the entire chromosome set. See, all are in duplicates. This is so many pairs of chromosomes, right? Maybe human section hai to इसमें all 23 pairs you will see, and a particular chromosomal region which is having trisomy is seen, and that will be useful in diagnosing the particular condition.